believe. I'll have to double check that. Yes, question. I'm sorry. I can't hear. So the question was about materials from the labs, and so there are lab handouts, and there may be materials that are presented in the lab itself. If there are any slides or so forth, those will be posted. If they haven't been already, they will be posted. But the questions are not going to be all a list of questions to define terms and things like that. They're going to be more synthesis questions where you put together material that you learned about in lecture and a lab to address them. So most of what you are learning about during a lecture and a lab will contribute to you being able to address those questions well. But you can also use the material from the class, the PDFs, including the slides, materials from the lab, your notes, so forth. The only thing I say is do not use other material. Do not communicate with other classmates about the exam questions or your responses to the exam questions. Right, so your information that you can utilize, everything you've learned about in class, text is a reference, your notes is a reference, and so forth. I don't have time to announce it today, but the other assignment that comes up, I think in mid-March, I have to look at exactly when this is due, but the proposal for the research summary assignment, that is open. Read about what the requirements for the paper are. Find the paper. You can propose it now. Already, several students have proposed a paper. And I think I've approved them all. If you propose something before the deadline, please let me know about it because I'm not constantly going there to check and I don't get any notification that somebody submitted their assignment. So if you submit a proposal and want me to evaluate it, let me know about it. And I should be able to immediately approve it or say, you need to find something else. One of the requirements for this paper is that no other student has chosen that particular paper. There are tons of papers out there, so I think I have not had it in prior class where I've had the same student choose a paper. But it can then be advantageous to pick one early, although there's so, you all have different interests. The chances that I think you'll pick the same paper are really low. All right. Class business. Lab this week will feature the groups that we're covering in the lecture this week, the Cnidarians and hopefully the Ctenophores. I've not seen if we've gotten the Ctenophores in. They can be rather fragile beasts, but we often do get some in and can observe them in the lab. So we talked about general features of Cnidarians and started going through the major classes of Cnidarians. We got through anthozoans. We're going to be covering the rest of the groups, rest of the classes today. Briefly talk about phylogenetic hypotheses of the Cnidarians, because it's important for understanding how traits have evolved within the group. Then we'll talk about Ctenophores and end up with a little bit of a discussion about sponges versus Ctenophores as being the most basal group of animals. At least that's my plan. We'll see if we get through it. Very quickly, if we have a few insights from students, something interesting you learned from the lecture reading, or from the text reading, the chapter reading, about Cnidarians or Ctenophores, or some unique experience you've had with them, something you've learned about in a different class, or elsewhere, Ctenophores or Cnidarians. Nothing. Yeah.
I think like Ctenophores are in colonies, like where they stretch out and they move via cilia.
So they can swarm. There could be huge numbers of ctenophores. You can see in that image on the lower right, they have these fused cilia that make up those bands, those rows that allow for them to move. They don't have great mobility, but they can move to some extent. They can swarm. And I'm not going to talk about it today, but there are cases of ctenophores that are invasive. They've invaded the Black Sea, and then have impacted fisheries in those areas because they're consuming food items of larval fishes and so forth. And so typical fisheries that rely on some of those fish stocks are suffering. So there's an interesting group. Thank you. Something more about ctenophores or other Cnidarian groups? Do you have to get more things? So some of your plans for lecture. Next week we're going to talk about echinoderms. Hopefully, when we get through today's lecture, we can talk about echinoderms. But you have a chapter reading on echinoderms. Think about something that's, whoa, that's really cool. I'm going to make a note of that. So when he asks about this stuff at the beginning of class, I have something to contribute. Or I remember that time I found a sea cucumber and it vomited out guts out of its rear end. Things like that you may have experienced. All right? Anyone else? Last call. Something interesting about Cnidarians or ctenophores you learned or you've had an experience with? Nothing. Yeah. One thing I learned in preparation for the lecture that I didn't know, in a 2020 study of some of the endoparasitic forms, myxozoans, identified or found that a particular species of myxozoans. These are parasitic forms. What do you know about parasitic forms? Often they've lost various features that are present in relatives. They're highly reduced morphologically. Myxozoans, for example, do not have a gut. One study found in 2020, reported in 2020, that they discovered a myxozoan that does not have any mitochondrial genome to it. And we know within eukaryotes, the vast majority of eukaryotes possess mitochondria involved with cellular respiration. This particular group of Cnidarians have lost their mitochondrial genomes. How do they function? I didn't read much further than that. I didn't investigate exactly what's going on in this particular case. But it represents reduction that we often find within parasitic forms. So, nothing more. Nothing more from the class. We discussed anthozoans. The next subclass we're going to be working our way through. We're going to skip myxozoans, and we're going to come back to them at the end of our discussion of the different classes. But the staurozoans are a marine group commonly referred to as stock jellies. They're not very large group. Anthozoans were very large, thousands of species. The staurozoans contain just around 50 species. They're referred to as stock jellies because they resemble attached medusae. It looks like a medusa with its mouth facing us and its tentacles emerging. That is attached. This upper part of the body is very medusa-like in appearance. We've only talked about Hydrozoans so far, but the medusa are the other body type, the other stage of the life cycle of most other cnidarians. They tend to occur in temperate, the polar, the relatively cold waters. There are some tropical species, but the greatest diversity is in temperate regions. They can be relatively abundant in shallow water regions. Relatively small polyps. There are some deep sea species, though, that are observed near hydrothermal vents. They were previously thought to represent true jellies, which are Skyfazoans, but have been shown to be members of their own class. Skyfazoans are, we haven't talked about them yet, we've only introduced them. These are the true jellies, or what we consider jellyfish. Their life cycle involves asexual reproduction during particular stages by which they can bud off small groups of cells that form these frustules. A frustule is kind of a term meaning a piece of something. These are cells that behave much like the larval stages of this piece. You can see it resembles the planula larvae that are produced by the adult medusans, the adult starozoans, through fertilization of gametes from male and female individuals. So the other case of asexual reproduction is as the starl polyp stage, early stage of development. They can produce frustules and ultimately produce other individual polyps. Single polyp again with the mouth in the central region. The starl polyp stage has a number of these arms. Subsequently develops into the adult, what is termed here the staromedusan stage, in which there's a number of tentacles associated with each of these arms. The larval stage of starozoans is similar to that of other cnidarians, planula larval stage. The difference is within staromedusans, these planula larvae are non-ciliated, and so they're not very motile. They're typically referred to as creeping larvae, because they'll ultimately settle onto the seafloor and then move, but not through the action of cilia. So they're capable of some movement, but not much. One thing I'd like to point out here is another unique feature of starozoans. It's illustrated this image on the left. Notice anything interesting about the coloration of that individual? How would you describe its colors? Yeah. Looks like it's glowing because of all those little white spots. It's got a lot of these little white spots, and those are unique to starozoans. We don't tend to see those in other cnidarians. These are little white spots that tend to occur along the arms. What those are concentration of cnidocytes. So nematocyst-bearing cells are concentrated in those white spots. The gonads of starozoans occur within the gastrovascular cavity interior to where those white spots are. And so some people have proposed that maybe those concentrations of nematocysts are there to protect gonads, but it's really not known what function they serve. It's something that is also unique to the staurozoans. We do not have any staurozoans to observe in the lab. They're really cool beasts. We don't have them. The rest of staurozoans are included in this clade here known as medusozoans, because again, this is assumed that this represents an attached medusoid stage, but the true medusoid body form is something that evolved based on this tree from the common ancestor of the hydrozoans, pubozoans, and skyfazoas. This is a free-living stage, and the stage within these organisms that is producing gametes. It's lost in some groups, it's retained in some groups, attached to the polyp stage, but vast majority of individuals within these groups possess a medusoid form. We're going to be focusing right now on aspects of hydrozoans, which is a rather large group, I think close to 4,000 species. (3,800 species), mostly marine, but with some freshwater species. And they exhibit a variety of different life cycles. Some are, like I mentioned a few minutes ago, some are only exist in the polyp stage. Others, such as this one here, trapuline hydrozoans, only exist in the medusoid stage. The larvae develop directly into medusae. There's no polyp stage. Because there's a lot of variation in the life cycle of hydrozoans. Many of the freshwater hydrozoans, hydra, for example. The gonads occur in the body wall of the polyp, and gametes are released directly from the polyp. Hydrozoans come in a variety of different shapes and structures. There are hydrozoans that exist as solitary individuals, but many are colonial. Start off as a single polyp. The polyp buds off another polyp, shown here, and then they're interconnected by a shared gastrovascular cavity. Here we have an example of a single polyp. Here's another colonial. This is a colonial hydrozoan. Anyone know what this species is? Yeah. Portuguese manowar. Have you seen these?
No, I wish.
No. Okay. Portuguese manowar. So you've heard about them at least. Portuguese manowar have a float, and otherwise is comprised of multiple individual polyps, individual medusae that form this entire organism. It's a weird beast because it's made up of different individuals, polyp and medusoid stages that form this. This is not a jellyfish. A jellyfish is a single medusa. This is a weird colony of polyps and medusae. What does that look like? Another type of cnidarian. There are some hydrozones that secrete a calcium carbonate skeleton. Have we heard this before? Like with corals. These are actually known as fire corals. The difference can be that what you can think about how a coral skeleton is structured. It has these internal scleroseptae. They're hexamerous. There's a six-fold symmetry. We don't see any of that within the fire corals. The typical life cycle of a hydrozoan, in this case a colonial hydrozone, species known as Lobelia, or genus known as Lobelia. Individuals began as a single polyp. Through asexual reproduction, they will bud off additional individuals, interconnection of the gastrovascular cavity amongst these individual polyps. And then there are feeding polyps, referred to as hydrant in our figure. They're also known as gastrozooids. Zooids typically referred to as types of specialized individuals in a colony. Gastrozooids are the feeding polyps, or the hydrants are the feeding polyps. There are other polyps, though, that are reproductive polyps. Gynangia, or gynangium, singular, as illustrated here. No, that's horrible writing. Oh, man. Or are reproductive polyps that bud medusae. These eventually break off from the gynangia and then develop into a medusoid, free living stage. Medusae are dioecious. So one individual medusa is either male or female, producing either sperm or eggs. Fertilization from these produces a zygote that develops into a ciliated larval stage that then settles on the colony. Think of the polyp stage that gives rise to the medusae. The medusae are either male or female. So what about the polyp stage? May they produce both male and female medusae? Or are they only capable of producing just one? Different hydroids do it differently. Some hydroids may be hermaphroditic, simultaneous hermaphrodites, as in terms of the polyp stage, in which they're capable of producing both male and female medusae. Others may be sequential, where in an early stage they're producing male medusae, in a later stage female medusae, or they may be dioecious, in terms of an individual colony of polyps is only going to produce either male or female medusae. So it's a lot of variation in the life cycle of these in terms of partitioning of male and female individuals and gametes. Remarkably diverse. the medusa of hydrozoans, the typical medusa, very distinct from the medusae, as we'll see, of other cnidarians that possess medusae. From your readings, do you remember any of these? What are some of the unique features of the hydrozoan medusae? One of the things that they possess is this inner shelf-like projection on the edge of the bell. Projects inward. What does it do? It essentially makes this area underneath the bell smaller than it normally would be. This particular shelf, here it is, a velum, like a veil. Little velum that projects inward. What it does is it changes that surface area, and so when muscles contract in the bell, it provides more jet propulsion than if it didn't have that shelf. This velum is unique to hydrozoan medusae. Otherwise, medusae have a mouth with lobes around the mouth. Other medusae often can have these oral lobes. It leads in to the gastrovascular cavity. And then there are typically 4 radial canals so if we're going to look at the bell of an individual, imagine the mouth is in here in the center, the entrance to the gastrovascular cavity. There are canals, typically 4 of them, that lead out along through the bell. See these here, radial canals, and you can see the holes for the others. That then ultimately connect the ring canal, shown here. So this is canal around the bell. So it's essentially, an organismal-wide stomach. These connections from the mouth into the rest of the bell of the animal. Gonads are produced along the ring canal. For this particular species that is illustrated, the polyp stage has a simplified polyp, and it too can produce buds, little frustules, that are again essentially an asexually produced larval stage because they're very similar in structure to the planula larvae. In terms of some of the diversity of hydrozoans, I already kind of introduced fire corals. Fire corals are hydrozoans that produce calcium carbonate skeleton. I was surprised to learn this because there's such a diversity of body types, but there are only 16 described species of fire corals. They make up a distinct family of hydrozoans. And they consist of, it's the group, the genus which they all occur in is known as Millipora. Lots of thousands of little holes, because the holes in the calcium carbonate skeleton are where the individual polyps emerge. Compare this to a Cnidarian skeleton, and it would be very distinct. You don't have sclerosepty present within the calcium carbonate skeleton. You just have these tiny pores where the polyps emerge. Like other hydrozoans, these do have a medusoid stage. Certain polyps will bud off medusae, and then it's a similar life cycle that we just saw for reveals. Except the only difference is these secrete a calcium carbonate skeleton. Siphonophores, I showed earlier the Portuguese man-o'-war, but there's a lot of diversity in siphonophores. Where do we put them? Over there. And again, these are a bizarre group. Of hydrozoans with around 170 described species, not all that large, but great morphological diversity. We do have preserved specimens in class to be able to look at. And these species, or these individuals, are developed, these colonies of individuals are developed from the budding of colonies, production of budding of polyps, production of medusoid individuals that all then serve a particular function. The float of a siphonophore. You see in some of these is a modified polyp, presumably. Quite enlarged polyp. And the different individuals, the different zooids, have different functions. This is the best illustration I have that attempts to show those individual components of Portuguese man-o'-war. What this is a cross-section, part of the float that contains the tentacles. And the tentacles are modified polyps, and then GG, the gonozoid forms, the gastrozoid forms that are involved in feeding. So these are, look at them, and they look like a single individual, but they're comprised of these individual polyps and medusoid bodies that are forming the entire individual. It shows the protozoid, proto first, the first zooid, the first individual. This is the larval individual that modifies into the polyp and then buds the rest of what becomes this composite individual. Another group of hydrozoans that don't really fit the bill of what we think of as a hydrozoan are these chondrochords, or porpetids. Chondrophore, we'll see phora, porifera, with an F sometimes even, it means bearing. Right, to bear something, to have, to possess. Chondra. Anyone know what chondra is? We haven't covered them yet. It won't be a while. Anyone here know what chondrichthyes are?
Sharks.
Yeah. So sharks and rays and skates and ratfish are chondrichthyans. Ichthyology, the study of fishes. Chondrichthyans. So they're fishes with chondr in them, right? The other types of fishes are osteichthyous. fishes. Oste. Anyone know? Osteology. What's osteology? Study of bones. Bony fishes are osteocles. Sharks. What kind of hard parts do they have? Yeah. Cartilage. They have cartilage. So chondrochords are bearing cartilage. They have these hard substrates to them. These upper regions of these blue buttons referred to, they're not all blue, and these by-the-wind sailors, they occur, these are species that are, again, like Phenophores, they're colonies of individual zooloids, but in these cases, Chondrochords are colonies only of polyps, but they produce a float. So they live at the surface of the ocean. These are the marine group. That form a unique community that's known as the Newston, as opposed to the plankton. And plankton are organisms living in the water column. The Newston are organisms, Neuston, organisms that live on the surface. And they do so by having these upper floats that are cartilaginous in appearance. This is a photo of a beach showing tons of what are these referred to as the by-the-wind sailors, because they have this little cartilaginous-like sail to them. They're capable of moving based on wind currents. When you get wind currents that are coming onshore, you may get mass stranding events of some of these chondrochords. Originally, they were thought to be closely related to Scythonophores. Not the case because of the similarity in terms of habitat and the fact that there's colonies of all these variety of different polyp stages. They're related to other hydrozoans, included, they're not in the same family, they're in the same order as birochords. In terms of their anatomy, again, this upper float, there are air pores, there are air tubes and sacs within it that allows it to be buoyant. And then we have modified polyps that are producing the tentacles, a gastrozoid that is associated with feeding, gonozoids that are associated with gamete production. Associated with what do gonozoids do? They produce medusoid forms. Medusoid forms then produce the gametes. But they're a really cool group, and oftentimes you can find these washed up, like you can see Portuguese manowar washed up. You can find these washed up to be able to examine their morphology, and they're quite a gorgeous group. The by-the-wind sailors have a very similar morphology. They're closely related, in the same family in terms of gastrozoids and gonozoids that make up the individual, and so forth. So similar morphology of the different types of chondrochords. And I know I wrote it down. There are, I don't know all 4 species, but there are just 4 species of these chondrochords. But they can be something that you commonly come across, at least when you're at a beach or doing Newston toes, the plankton toe, but on the surface of the ocean. The final group I want to talk about of hydrozoans are the tracheomines. Hydrozoa is a class that contains just 2 subclasses. Tracheomina is one of them. Hydroid Hydrozolina is the other one. All the other hydrous ones we've talked about so far are in Hydrozolina. It contains vast majority of species, traculines. It's about 165 species, so this contains, how do we say, more than 3,600 species within a vast majority of these. But the traculines are interesting because they typically have no polyp stage. There are some that do, but most will undergo embryonic development, Colonial Larval Stage, and ultimately directly develop into the Medusoid stage with no intervening polypoid stage. So that's a survey of some of the hydrozoans that they are. The next major group to cover, the next two major groups are the Skyfazoa and the Cubozoans. And you can see in this tree, they're indicated as close relatives. Indicates here that polyp is reduced or lost. In many cases, there are polypoid stages. They're not just well known. Another feature that is shared by these is a rhopalium. A rhopalium is a unique structure associated with the Medusoid stage that is essentially a package of sensory organs. There are balance sensing organs, statocysts. There are eye spots. In some cubozoans, there are image-formed complex eyes that occur in these rhopalia. The unique feature of the Skyfazoans, strobilation, formation of a strobila, or literally a pine cone. These are how Medusoid stages are produced from the polypoid stage. And these are the true jellies. Variety of different shapes and colors and forms. We should have some of these upside down jellyfish in the lab this week. They actually will incorporate unicellular eukaryotes, photosynthetic unicellular eukaryotes, into their body to be able to photosynthesize, and they live in upside down fashion. Cassiopeia. Yeah.
I had a clarifying question.
Uh-huh.
So these are true jellies. Then there's jellyfish in hydrozoans and stratozoans, and then there are also Ctenophores. Like, what is a jellyfish? Is that just any animal that floats in water?
Oh, that's horrible. So the question is, what the hell with all this jellyfish? And what is a jellyfish? Is a medusa? Some people would refer to that. If somebody said the jellies of the sea, they may be including all medusoid forms as well as ctenophores in that description. The jelly, right, is in reference to the fact that they have this extracellular matrix between their epidermis and gastrodermis, the mesoglea, that gives them their jelly-like appearance or structure. Ctenophores also have a similar internal structure, internal mesoglea. They're jellies, and that's one way of describing them, as jellies.
It's like, because like, think of like penguins, and then you think of all the different species of penguins, and they're all in the same class, right? But
Right.
jellies are not.
They're not, and so you would, if it was me, because it would be a polyphyletic assemblage, you would say jellies like that, because it's not a true taxonomic group. Why we think of these as true jellies or true jellyfish is because they're a monophyletic group that is distinct from the medusoid forms of other cnidarians. So, yeah, when a zoologist is talking about a jellyfish, what they mean are Skyfazoans. Otherwise, they'll say the medusoid stages of hydrozoans and so forth. So it's a semantic question, but a good one. Skyfazoa, skypho cup animals. most predominant in their medusoid stage. They're all marine. They're largely, at least I don't know any exceptions, dioecious. So you have male and female medusoid forms. And their medusae are distinct from those of hydrozoans. What was one of the unique features of the hydrozoan medusae? Yeah.
They have like that inner jump on their under the shell, right?
Exactly. They possess the velum, that inner thing that helps with movement, better jet propulsion. We do not have, this is the edge of the bell here, the edge of the bell here. There is no inner shelf-like material within the medusae of Skyfazoans. What else is unique about Skyfazoans? Their life cycle. Again, talked about strobilization as a distinct feature of this group. We start off with zygote that develops into a ciliated glycosyl larvae, like most Cnidarians have, except for starozoans. Final settles. It forms this polyp-like stage known as Skyfazoa, a cut mouth. It can bud to produce additional polyps, a common theme that we see in other Cnidarian groups. But at one point, it will start to form these plate-like structures, and it doesn't really look like a pine cone to me, but that's what a strobila, the plant biology term for a pine cone is a strobila. It forms these strobili, in which all these different layers are the young medusoid stages. Once they emerge and are individual, they're known as nephyri, singular nephira. That's a small, immature medusoid form. So it's very distinct from the budding that occurs in those reproductive polyps. Reproductive polyp of a hydrozoan buds off medusoid. This one forms them in these layers as shown here. The medusa is distinct from that of other groups. One is, we already talked about, you don't see it here because the velum is absent. There's no inner shelf on the bell of the medusa. The oral arms, extensions from around the mouth, are usually quite exaggerated within the medusa of Skyfazoans. There's a ring canal around the bell. Remember, in the hydrozoans, that ring canal is connected to the gastrovascular cavity through radial canals. There are radial canals here, but they are highly branched. It's kind of shown in this image. You can't really see that here, but highly branched. And they emerge not from the central vascular cavity, but from these gastric pouches. So there are also gastric pouches from which the radial canals emerge to connect with the ring canal. As I mentioned in that tree figure, something unique to Skyfazoans and their sister group, the Cubozoans, that we'll get to shortly, are the presence of these structures known as Rapalia, that house various sensory structures. Statuses that are balance sensing organs. the eye spots. And sensory bits. Again, if we think about a bilaterian, head, cephalization, concentration of sensory organs in one particular spot. How would you do it if you're a radially symmetric organism? You would have them distributed around your periphery. This is a cool little question.
Can you just repeat what ropalia is referring to?
A ropalia is this structure that occurs along, different areas along the edge of the bell of the medusoid form, and it is a part of the body that houses various sensory structures within. So this is a nice little video that kind of summarizes aspects of the life cycle. You could also see aspects of the anatomy and morphology of these individuals. Let me just a few minutes. Wow. Moon jelly, commonly wash up ashore on Gulf of Mexico beaches. I remember that.
Moon jellies begin their lives as tiny polyps anchored to the seafloor. They are ghostly forms of life, no larger than the tip of a pencil. In fact, just a few times each year, moon jellies undergo one of the most fantastic transformations in the animal kingdom.
At some point, those polyps will begin to divide. And it's almost like they form little plates, one on top of each other.
Each polyp forms dozens of orange plates. Each plate in turn becomes a single animal called an ephyra.
The ephyra can be considered the juvenile jellyfish stage, each of which breaks off from the pulp stem and swims away.
Over the course of a month, the juvenile jellyfish feed on plankton as they develop into adults.
See if you can see the four gastric pouches.
Fully grown, they can reach nearly 2 feet in width. When they spawn, these jellyfish gather in massive swarms near the coast. Males cast threads of sperm into the water. Collecting the threads with their frilly arms, females capture and ingest the sperm, which then fertilize their eggs. But their offspring will take root as tiny polyps and start the cycle anew.
I think they said it, I don't think I said it either, but it's to those, so within Hydrozoa and Medusae, the gonads typically form around the radial canals. Within the Skyfazoa, the gonads form within these four gastric pouches. And as you saw, sperm are released by the males and picked up by the females, and fertilization takes place, and then the embryos are released by the females. All right. Second to last group are the cubozoans. They're related to scyphozoans. They also possess these Wopalia, sensory structures. And it seems like it's cheating, but they possess a valerian, a velum, unique to hydrozoans, veil, valerium, same root. It's a similar structure. Cubozoan medusae have this inner shelf-like part to their bell that aids in movement. It's missing in Skyfazoans, so one hypothesis would be, well, a velum evolved here, and then Skyfazoans lost it. I don't know why they want to lose it. It seems like it's advantageous. But Skyfazoans, the ancestor of all Skyfazoans, lost that particular trait. That would be one hypothesis, right? Two steps involved in that. We gained it in the common ancestor of all, and then lost it here. Big question.
I guess it isn't always like when you're making a phylogeny, you want the least amount of steps. So why did they do it twice when the least amount of steps would just be the Scythozoa and philosophy?
That's a good question in terms of phylogenetics. If you were basing it on morphological features, you'd want to have enough to make as few changes in the character states of all of those. Some of those would end up having multiple states. But like with this tree, I don't know how the authors of our textbook comprise this tree. And there's some parts of it that are wrong based on our modern thinking and relationships. But another way to do it is build a tree with molecular characters and then plot morphological characters. But good. But either way, it would be the same number of steps because we could have it evolve here in the common ancestor of all. So one step, and then lose it here. 2 steps. Or we can have it evolve here and there, which is 2 steps. So which, if we are building this tree based on that character, either way requires 2 steps. The hypothesis that I gave that it evolved here and then was lost in Scyphozoans is very unlikely because the valerium in cubozoans is structurally distinct. There are a number of features about it that are different than the velum that we find in hydrozoans. So that supports the notion that it evolved twice because it's not identical. It doesn't have similar structure in both of these groups. Cubozoans are known as box jellies. Cubo, or because of their cube-like shape, their box-like shape. Like shown on the tree and discussed for scyphozoans as well, they tend to be most known for their medusoid stage. But most go through a polypoid stage. It's not just well described for each. You can imagine a medusoid, pretty conspicuous. Some of these polyps are rather minute. And so determining that hey, this is a polyp of a scyphozoa or a cubozone may be very difficult to do. But the general structure of the medusae of cubozones is distinct from that of other Cnidarian medusae. This box-like nature. Look at it in cross-section from above, very box-like. It's got these 4 corners to it. And unlike medusae of other Cnidarians, the tentacles emerge from these corners, and there's this thick part at the beginning of them, referred to as the pedalium, that gives rise to the tentacles. So morphologically, they're distinct. These figures don't identify it, but this is that valerium that's analogous, because it's independently derived, to the velum of hydrozoans and performs similar type of structure. They are similar, though, to scyphozoans, as shown on that tree, in that they too possess these structures, Wopalia, that have statuses, sensory pits, eye spots, And in some beasts, you can actually see them here. So I get these images over here. Actual pigmented, highly pigmented, image-forming eye. I've never looked through the eye of a cubazone, but apparently they can form images. Again, they're distributed around the periphery of the individual medusa. So the various features that are unique about these medusae. They're all marine. There are only, I saw that there are 48 described species. Some of these can be deadly. Mentioned on Monday, beaches are often closed in Australia when there are swarms of cubazones, the dangerous ones that are nearby. There are box jellies that will come ashore also in the Hawaiian Islands, but they're not as bad. But I do know swimmers in Hawaii will often send notifications out when there are cubazones in the water because they don't like getting stung. They won't kill you, but they don't feel good. The life cycle of a cubazone is similar to that of other cnidarians. Again, where we know they go through a polypoid stage. The polypoid stage, this is an early study of these that caught the polyp stages of a cubazone. They can undergo budding to form additional polyps, so they can undergo asexual reproduction. But then, the single individual polyp undergoes metamorphosis into a single medusoid form. So it's not like in hydrozoans where a polyp, reproductive polyp, is budding off numerous medusae, or a Skyfazoa, where we have the strobila that is producing numerous aphyri that are going to then develop into medusae. Individual polyp, although it can produce additional polyps, that polyp is only going to form one medusa. I know there's an exception, a recent study that suggests the polyp may leave a small bit of tissue after it undergoes metamorphosis, and that could then transform into a new polyp. But that's only known for one particular case. But a unique formation of medusae for cubazones compared to the other types of cnidarians. Budding versus strobilation versus complete metamorphosis. All right. Final group, myxozoans. These are the endoparasites of fishes and worms, and it's not included within myxozoa, but another endoparasitic form, Polypodium Hydroforming, that appears to be a close relative of the myxozoans. They both are cnidarian, though, in that they possess nematocysts, or highly modified nematocysts in terms of the myxozoans, that are, at least for the myxozoans, associated with getting into the host. The nematocysts of myxozoans in this infective spore stage here are known as polar capsules. Originally not called nematocysts because these were thought to represent unicellular eukaryotes, protists, and not cnidarians. What we find, if you sequence a bunch of myxozoans, they group firmly within the cnidarians. And prior to molecular data, myxozoans were not inferred to be cnidarians. Only once molecular data showed that it grouped firmly within cnidarians, we'll be able to look at the polar capsules and say, hey, those represent nematocysts. The life cycle, the involves parasitic stages that occur within multiple different types of hosts. Salmonids, for example, various fishes. There are also the amphibians. There's some reptiles, or mammals, and birds that may be an intermediate host of mixozoans. So there's maybe some mixozoans that are purely terrestrial, but many are freshwater. A mixospore emerges, and this is where you can see the polar capsules, may be consumed by an annelid worm. We'll talk about annelid worms later, but this includes earthworms and so forth. Be digested by the worm, and then the polar capsules, the metacyst, fire, and allow the mixospore to go through the gut wall. Actinospores emerge from the worm host, develop within the worm host, and then infect salmonids. And it does cause whirling disease in salmonids that can be detrimental to fishes, especially when they're in a fishery. They're highly reduced. They don't have any nerves, any muscle, no cilia that we find within other Cnidarians. And again, that one species I mentioned at the beginning of class that has lost its mitochondrial genome, which I want to read more about because I find completely bizarre. Polypodium Hydroforming, which is the other branch on that tree. It's only one species known of this particular group, and it infects Skyfazoa for you fish people out there, sturgeons, and paddlefish, freshwater fishes. There is these weird stages, stolen stages or tentaculate stages that emerge from the fishes, and then these divide to form these tentaculate stages that are reminiscent of the medusoid stage of the Cnidarian. They possess nematocysts. These stages then deposit a gametophore on fish larvae, and then fishes are infected and later give rise to the stolen stages. So these, like showed in the tree figure from the text, appear to be closer related to the mixozoans, which suggests a common ancestor that evolved this parasitic life cycle. In terms of phylogenetic relationships, this is 2018, most recent evaluation of the relationships with the Cnidarians. It shows slightly different relationships than what we see in the text tree, so I just wanted to point this out. They looked at a variety of different Cnidarian groups, sequenced various genes from them, and then reconstructed a phylogeny. And we talked about the anthozoans being most basal, that's supported by this phylogeny. Octocorals, Cnidarians, hexacorallians forming distinct groups. Colectinians are the corals, Cnidarians are sea anemones. These are other types of anemone-like beasts. Polypodids and mixozoans form a distinct clade that they're referring to as Endo-Hydrozoa because of the endoparasitic life stage exhibited, life cycle stages exhibited by these beasts. And this is the other clade we look at. Hydrozoans, monophyletic Skyfazoans, and Cubazoans are closely related species, closely related groups, sister groups. Rhopaliophora, very good name for that particular clade. It's Sarazoans off here. So how we think about classification of Cnidarians is as I've been presenting in class. But if we look at tree the text is given, and the tree that's given in this recent analysis, how do these differ? Yeah, you see it already. Good for you.
Just a really small thing is the tree from the textbook isn't as specific, and it doesn't include a breakdown of the different kinds of Anthozoa.
Oh,
The
that's,
other one does.
what about the topology? Anyone notice anything about the topology? Yeah.
Stars are more closely related to Hydrozoans.
Very good. So it shows that, here we have Staurozoans being the most basal member of this Medusozoa clade, right? Here, the most basal member of Medusozoa are the Hydrozoans. Where did this tree said that Medusoid forms evolved in everyone but the Staurozoans. This one would imply that evolved here. And so Staurozoans are indeed just attached Medusae, or they lost the Medusoid stage and only exist as a polypoid stage. Differences in tree topology require us to reevaluate how characters evolve and group. Sometimes it can be quite informative to see a tree and understand how these traits have evolved. Good catch. All right, now I have a few minutes left, but I think I can introduce the Ctenophores. Our text doesn't have lots of detail on them. Ctenophores is a phylum, only about 200 species. They're only marine. And they're, like we've discussed already, they're similar to Cnidarians. They exhibit this primary radial symmetry. They exhibit just two embryonic tissue types. They're diploblastic. In between these layers is a mesoglia, this jelly material, this extracellular matrix that is produced. It's mostly acellular. There are some cells in the mesoglia of Cnidarians and Ctenophores, but not lots of cells embedded within. Like Cnidarians, they also possess muscle and nerve cells. They're predatory. They have a mouth. Some eat other Ctenophores. They have mouth. Some have lobes. Many have tentacles with sticky structures on them that allow them to capture food. They show a variety of different morphologies. At the very beginning of class, I showed a movie on Ctenophores that showed some really bizarre morphologies with these lobes kind of greatly extended, but kind of similar, reminiscent of this piece. Some of the Ctenophores that we're hoping to get in the lab have no tentacles and should look like these. Ctenophores are also known as sea walnuts because some of them have shape kind of like a walnut. Vast majority live within the water column, but there are some. This is a benthic individual that lives on the sea surface bottom. These are rare. I've never seen one, but few species have a benthic habitat. In terms of their generalized morphology, they have a mouth and gut that opens into numerous diverticula, and then there are anal pores on the opposite end from where the mouth is. You notice these are referred to as anal pores and not an anus. For a long time, it was thought that Ctenophores have just a blind gut. Food goes in the mouth, and waste products come out of the mouth. But it was subsequently discovered that they actually do pass food all the way emerging from their anal pores. This is a study just recently from 2016 that I could show a movie of a Ctenophore pooping as one of the last things in class today. But it shows, what? I need to turn that off. But it's time-lapse movie actually of a ctenophore defecating. So there's food particles that are emerging here. This is the aboral end of this particular ctenophore. Looks like there's some kind of processing going on in the material, and then it ultimately emerges. So we talked about cnidarians don't completely undergo gastrulation. Ctenophores, too, consider not to undergo gastrulation. So these anal pores are a connection between the digestive tract and the aboral and rear end of the beast. So we saw a ctenophore pooping. But this is important because previously we thought they just had a blank. They possess various muscles throughout their body, muscles associated with the tentacles that allow for the tentacles to form, muscles around the digestive tract, presumably related to movement of food within it, and various other muscles throughout the body. Muscles. Nerves. They exhibit a nerve net, no centralized nervous system, similar to what we saw in cnidarians. Nerve net, various nerves associated with tentacles, and some even within the mesoglia. There's various staining showing the nerves, nerve nets, mesoglia nerves within the body of the tentacle. Ctenophore, I forgot to mention this. Ctenophore. It's a C-T-E-N, teen. That means comb. They're comb-bearing beasts. They have these 8 rows of plates of cilia, fused cilia that occur in these different lines. One of the unique features of the ctenophore, what it's named after. They have nerves. Anything that they have that are somewhat similar to nematocysts. So all it is to put ctenophores and cnidarians together in a single group because they all have radial symmetry, primary radial symmetry. And ctenophores had these colloblasts that are essentially structures that can emerge from cells that then are used for prey capture. They're not injecting any venom. They're just got sticky particles on them, mostly present within the tentacles of the tentaculate species. Ctenophore life history is not all that interesting compared to cnidarians. They mostly undergo direct development. They, in terms of their cleavage patterns, it looks like deuterostomes in terms of radial cleavage. There's 8-cell stage here. The cells are overtop each other. They develop into embryonic stage that more or less resembles the adult stage. They're names for the larval types, but they're kind of like small versions of the adults. Fossil record. Sponges go way back. Ctenophores do go back into the Cambrian. And most interestingly, some of those Cambrian individuals, these date back to the very lower part of the Cambrian, have actual hard parts, skeletonized parts, these rods and so forth that are inferred from the fossils. And the fossilized species are over here, the extant species are within here. We have 2 main groups of ctenophores. One large group that is made up of tentaculate forms and the burroids that are typically the seagull nest, thought of that are atentaculate, have no tentacles either. All right. I got 3 ctenophores, but I didn't talk about this. We'll discuss this at the beginning of lecture on Monday. So think about sponges and think about ctenophores. And think about why one group or the other makes sense as being most basal, and what it means if ctenophores are basal or sponges are basal. Question?
Yeah. Because you've been highlighting this species, and